Hereby, I open this academic ceremony in which Mônica Maurer Sost will defend the academic thesis, A Journey into the Gut Bacterial Profile and Functionality in Obesity and Health, from Dietary Fibres and Polyphenols to Sweeteners. May I invite you to present a summary of your study and the conclusions of your thesis.
Thank you very much. Dear co-supervisor, supervisor, dear assessment committee, dear friends and family who are here and who are online giving me support, I would like to present a few results of my thesis. So you might have seen or heard this statement, "We are what we eat." And this is actually a title of a book written by this chef, Alice Waters, where she says that every decision that we make about what we eat affects not only our bodies, but also our health and the world in general, so all our communities and our environment. One important health condition affected by what we eat is obesity. In 2022, more than 1 billion people in the world were living with obesity, which results from a combination of genetic predisposition, behavior factors, and environmental influences, including lifestyle factors like our diet and also physical activity. And when we choose an unhealthy eating habit, combined with a reduced physical activity, we can cause an energy imbalance in our body, leading to weight gain. A key strategy for treating obesity involves adopting a healthier eating pattern and increasing our physical activity so we can balance energy intake and expenditure, leading to weight loss. But then, what are we have been eating that the numbers of people with obesity have been rising so much? Well, currently, most people eat what we call a westernized diet, which contains low number of fruits, low number of vegetables, legumes, and whole grains. So that means low amount of dietary fibers and polyphenols. And they also end up eating a high number of animal-based products like red meat, sugar, and highly processed foods. While the general dietary recommendations are that we should eat a diet rich in fruits, vegetables, legumes, and whole grains, so full of fibers and dietary polyphenols, and low in meat, simple sugars, and low in highly processed food. And we call this kind of diet a plant-based diet. But how exactly the food that we eat affects us, and what happens with the food we eat? When we eat a meal, our body starts the process of digestion, which is basically breaking down the structure of the food we just ate to smaller pieces so our body can absorb and use all those nutrients we ate. And we are able to break down the food because our body produces some tools to help us with that. They are called enzymes. However, our enzymes cannot break down everything we eat. Food components such as dietary fibers and polyphenols will reach the colon, passing through the whole system of digestion, basically intact. In the colon, we find a whole universe of organisms, our gut microbiota, which is a complex and highly individualized ecosystem consisting of a diverse array of microorganisms, including bacteria. And that's what I'm going to talk a lot today. And luckily us, these bacteria they are full of enzymes. And those enzymes are enzymes that we don't have. So they help us to digest those food components, the dietary fibers and polyphenols. However, if we eat an unhealthy diet, the substances produced by the gut microbiota, when they break down the food we just ate, can be detrimental to health. While if we consume a healthy diet full of fruits, vegetables, legumes, so full of dietary fibers and polyphenols, we are consuming this diet that's very healthy and rich, and the substances produced by our gut microbiota will be also healthy for us. One example of these substances are the short-chain fatty acids. The short-chain fatty acids interact with our whole body and system, which beneficially impacts the function and metabolism of our body. So over the past 2 decades, increasing evidence has implicated the gut microbiota and what we eat into the pathophysiology of obesity. Thus, the aim of my thesis was to investigate the impact of various foods components on the modulation of the gut microbiome composition and function, with the aim of conferring benefits to the host, including both healthy individuals and those with metabolic impairments. And today, I'm going to present the results from chapter 2, 3, 5, and 7. Okay, but I thought I'd talk about bacteria, column, and food, but what exactly I did during my PhD, and how did I study this complex structure. Basically, what we did was, in our lab, we have this system where we use to mimic the column. Why that? Why is that? Because studies with humans are quite expensive and require ethical constraints that can be quite long to get rid of. But we have in our lab this in vitro model that mimics 4 columns at once. So each unit that you can see in the image can mimic 1 different column at the same time. And this is called the TIM2 system. So each unit mimics 1 column. I'm just highlighting one of them here. And with TIM2, we can mimic body temperature, column pH, also the bowel movements, gut microbiota profile, and also we can measure the substances, so for instance, the short-chain fatty acids produced by the gut microbiota. And we can test different foods and food components in TIM2 as well. So for chapters 2, 3, and 5, we use a pooled fecal sample to inoculate TIM2, and the experiment lasts for 3 days. So in chapter 2, I tested a traditional crop from here, from Limburg, the asparagus, green and white, and its effects on the gut microbiota of individuals that were metabolically healthy and compromised. We tested a fermented version of asparagus, but also we test those parts that usually by the client, so who buys the asparagus in companies, but also who buy in the supermarket, so us, get rid of. So for instance, the asparagus white, we have to peel off, and those materials go just for the waste. So my idea was to find something nice about asparagus and bring back to society some nice information so the growers can also take advantage of that, not only who consumes it. So my results confirm once more that the gut microbiota of individuals metabolically compromised is different from that of healthy donors. and that they also respond different to each tested product. We also saw that the gut microbiota from healthy donors produce higher levels of short-chain fatty acids compared to the metabolic compromise, in agreement with previous research as well. And we relate these benefits with the short-chain fatty acid production within the asparagus products with the dietary fibers that are present in those vegetables. In Chapter 3, we evaluate the effects of 2 different doses of citrus fruit extract rich in polyphenols on the gut microbiota functionality. After continuous supplementation of TIM2 with the citrus fruit extract, we saw a higher production of acetate compared to the diet without the citrus fruit extract. And this diet that didn't have the citrus fruit extract was basically a diet mimic a westernized diet. In Chapter 5, we tested the effects of 3 sweeteners on the gut microbiota of donors with obesity. Non-caloric sweeteners are used to replace sugar and decrease the caloric intake of individuals with obesity. But little is known about the effects on gut microbiota in that population. So we used them to test their effects. Sweeteners, they modulated the gut microbiota, and also we saw some responses regarding the short-chain fatty acid production, where sucralose led to the higher production of short-chain fatty acid among the sweeteners. So in this chapter 7, we use a slightly different approach, because evidence from human trials shows that not all individuals might have an identical response to the same food products or compounds. Thus, in this chapter, we use TIM2 as a screening tool for a personalized approach. So our goal was to find a dietary fiber mixture that would lead to the highest production of short-chain fatty acid for each fecal donor, so for each participant in this study. So this time, the TIM2 experiment mimicked the whole column, and it was used as the initial part of a clinical trial. So each volunteer was instructed to donate fecal samples that would be added to TIM2, one at a time. So for instance, the fecal sample of donor 1 was added to all units of TIM2, and in each unit, we added a different dietary fiber mixture. The differences between those mixtures were the food source and their combination. And with that, we found what mixture was the gut microbiota of that specific person responded best by evaluating the short-chain fatty acid production in the distal part of the column. And what we found was that the total short-chain fatty acid production varied among participants for the same mixture. So here we see that there are some people that barely respond and some people that had higher amounts of short-chain fatty acid. And then for mixture 2 as well, different responses, also for mixture 3 and 4. But we also saw that the response was different within participants for each mixture. So we can see here 2 participants that respond completely different to the 4 mixtures of dietary fiber. And this is the result for all participants. So they just respond as they wish. So the conclusions of my thesis was the asparagus-derived products, particularly the non-edible parts, so the peels that we just usually throw away, and the citrus polyphenols extracts enhance short-chain fatty acid production and support beneficial bacteria population. And we also saw that different non-caloric sweeteners have a distinct effect on microbial composition and short-chain fatty acid production. In chapter 7, we saw a substantial intra- and inter-variability in short-chain fatty acid production. And this highlights the personalized nature of microbial responses to dietary fiber mixtures. And we need still further research to validate the results for these in vitro studies with a higher number of participants, but also applied to clinical trials. And with that, I end up my presentation and give back the word to the pro-handler. Thank you very much for your attention.
Thank you very much. We will now start with the opposition, and the opposition will be opened by the chair of the assessment committee, Professor Penders, professor of metagenomic epidemiology and intestinal microbiology at this university.
Thank you. Dear candidate, first of all, I would like to congratulate you with your very nice thesis, which clearly shows a lot of hard labor and perseverance, and sometimes also under challenging circumstances. So congratulations for that. And I would also like to extend my congratulations to your supervisors, or Team Mônica, as you call them yourself. And I also liked, besides the content, also the cover a lot. So I was looking at this cover and I recognized a lot of items: the citrus fruits, bifidobacteria, flagellated bacteria. But I also saw some things that I not immediately recognized, or that I thought were maybe viruses or maybe protozoa. And I was wondering, is that indeed the case, or is that just my imagination? Are there also some non-bacterial microorganisms on there?
Thank you for your question and nice words, highly esteemed opponent. So my idea was to relay on the fact that as we don't know much about the universe, and we still have a long run to understand what is out there, we also do not understand completely what is the gut microbiota and the universe inside of us. So that was my whole idea. Although some pictures might, some figures might rely on the format of other things than bacteria, was more a ludic way to transport people to this universe that I work in the last few years.
Okay, so it's also an outlook to future research then.
Yes.
Okay, nice. Then I would like to discuss the results with you, and I would like to focus initially on chapter 2. And here you showed that the Aspergillus-derived substrates mainly modulated the metabolically compromised microbiota, enhancing diversity, shifting some key bacterial taxa, while the metabolically healthy microbiota remained relatively stable.
Yeah.
And you attributed this, amongst others, to differences in baseline microbiota composition between these two groups of donors, both in composition and functionality, as well as to the potential greater capacity for improvement in metabolically compromised microbiota. However, when I look at the results, I also noticed something else in the metabolically healthy group that made me wonder whether that could explain the effects. And I would like to take a look with you at figure 4a on page 49.
Yes.
And here you show the principal coordinates analysis with different beta diversity indices. And what I noticed was that the baseline microbiota differs between different experimental conditions.
Yes.
So at T0, not all of these microbiomes are the same, while it's the same donor pools, and there's no substrate added yet. So is this something that could explain these lack of effects, and could that impact your conclusions, you think?
So what we saw in this picture, I'm showing both groups together. So definitely there is a difference between the good microbiota profile at the beginning of the experiment between the metabolically healthy donors and the metabolic unhealthy. And I think that's is something that we have been working on, in the sense of my wish was trying to give asparagus to those bacteria in the good microbiota of the unhealthy and try to restore for a more healthy pattern. But indeed, some difference we can also see here, that the microbiota of people with from healthy donors, they were much more stable. So they didn't change as much as the gut microbiota of people that had some metabolic impairments.
Mm-hmm. But do I understand correctly if I look at figure 4a, for instance? Mm-hmm. This is the microbiota of metabolically healthy donors, right?
Oh, yes, sorry.
And if you then look at T0, for instance, if you look at the SEAM T0 samples, they start at a different point than the FA green, for example.
Yeah, so we had some concerns about timing of TIM2. So there were some experiments that took longer to be redone because some error or some problem with the system. So we had to stop the run for the specific groups, and one of them were the groups of the control. And then, indeed, we had differences, responses, I believe, although some people claim that the gut microbiota wouldn't change over time that much if we keep in -80. I think there are some modifications in this sense, and that would explain the beginning, the baseline being different.
So you think that it's the longer time that these samples were in the freezer that is explaining the shift in the composition? Okay.
Yeah.
And if you would redo these experiments, what would you do differently in the model or in the experimental setup to maybe control for this?
So I believe, although it would be more expensive, but I believe having triplicates instead of duplicates would enhance the results and the replication of the results in this sense, because then we can work with a main product instead of just a duplicate that can go either way if something goes wrong. And then we don't have this time being implicated if we have to repeat it. So I think adding one more replicate to the system would be helpful.
And would that, in your opinion, also really make you could it come to the conclusion that it's really the shift in the microbiota during storage time, or could you maybe, because what is T0 in this experiment? At what time is T0? Is that right when you start up the TIM2
No,
run?
so we had the adaptation period for 80 hours, so 16 hours adaptation, and then 2 hours starvation period, because over that time we are feeding the model with our standard, so a westernized diet-based media. And after that period of starvation, then we believe that the gut microbiota would have used all the resources that we had added beforehand, and then would be willing to start fermenting whatever we add to the model again.
So there is already a whole period of 16 hours where a lot can happen, where you don't have any information on. So would it maybe be an idea to measure already at inoculation?
Yeah, definitely. Yeah. Definitely. What is also, I think, a good practice, and that would be nice for future studies, is taking a part of the fecal sample with slurry to evaluate the sequencing and microbiota profile even before starting TIM2. So right away when we do this slurry, do the fecal sample pool, then we take a small amount to evaluate that. That would be a nice check.
Okay. I thank you for your answers. I give the word back to the pro-rector.
Thank you, Professor Penders. The opposition will be continued by Dr. Hernandez, Doctor in Food Sciences and Food Technology at the Institute of Food Science Research in Spain. Thank you for coming to Maastricht.
Thank you, Prorector, dear candidate. I want to congratulate you for your thesis. I think it's a very beautiful work, and also extend this congratulations to your supervisor, to your team, Monica, I just heard. So I have just a few questions regarding your work. It's mainly in the methodology.
Yeah.
So my first question is, on page 39, in figure 1, you present in vitro modeling that you will use in the whole thesis. And I realized that you have a kidney using peristaltic and dialysis systems. How does it work? Do you really think that you can mimic the kidney in the system?
So, well, yeah, it's kind of a push, but thank you for your nice words and your question, esteemed opponent. So we have in TIM2 what actually differentiate TIM2 for other in vitro models that mimic column. We have this membrane that mimic in such few ways the system of the renal system, the kidney system. So the substances produced by the gut microbiota will not accumulate that. So we mimic absorption of those metabolites over the whole process. And that is one of the features of TIM2 that differentiate most TIM2 from the most in vitro studies.
Yeah.
So it's not like a literal translation to the kidney system, but it helps to be more physiological in this sense.
What do you think if I challenge you and I tell you that I think this is not a kidney, but it's just the absorption of the small intestine? Yeah. What do you think about that?
No, I agree. I agree with you. I think it's mostly the absorption through the epithelial cells in the column than the process of the kidney system. Yeah.
Yes. Another question that I have is on page 125, you describe your experimental setup for the Sweetener study. And you said that the doses is expressed in milligram per kilogram of body weight, right? And translated to 100 kilogram individual. Technically, if I'm wrong, just let me know. Technically, the colon of the human is bigger in volume than the TIM2, right?
Yes. Yeah.
So how you translate this milligram kilogram body weight to the TIM2? How did you do?
So we just thought about the highest level recommended for using sweeteners, so the dosage, based on the weight of the donors. So most of them, the range was around 90 to 100 kilograms, and then we try to translate in the same to the scalability of TIM2. So we do not overdo the dosages. Okay.
Regarding your study about sweeteners, in your discussion, the general discussion, you noticed that I noticed that there was no control arm that combined the sweeteners with the fibers or carbohydrates that you were using. Do you think that would be important or not?
Yes. When we were setting up the methodology, we thought in two ways. So we have the 4 units of TIM2, so we could run 1 control and 3 treatments. And we thought what we really want to see. So because there is a lack of understanding on the action of the sweeteners by their cells, the idea was to provide just a sweetener without any or not much carbohydrate together and compare with just the normal diet. But I do agree that it would be nicer to have this positive control. And then maybe with a 5th unit we could run all in parallel together. That would be nicer. Yeah.
Because technically one of your conclusions said that during this experiment you see an increase in methane-producing taxa. So eventually, if you do this positive control, maybe you can reduce this methane taxa. Without results, you cannot say, but what do you think about that?
So I believe it would be possible to reduce, because then we have maybe the good microbiota will choose to fermented dietary fibers or the carbohydrates in that media instead of right away going to the sweeteners, and then the fermentation will be much slower. But because they do not have much choice, since there was just sweeteners, probably the fermentation is quite fast, producing a lot of gas rapidly.
And my last question is regarding analytical chemistry. You mentioned that it's very important to do other analysis like metabolomics. Talking about sweeteners again, imagine that you do this mixture. How important do you think is this metabolomics? Because these sweeteners, the structure, they are not completely degraded by the microbiota, some of them. So why do you think it's really important to do this kind of analysis, metabolomics, for example?
I think then we would link also which enzymes and which bacteria, strains or species or strains, but hopefully strains, are involved in that degradation. And then maybe we can, in the far future, decide which sweetener to provide to a certain person that would work better. That I think it's one way to look at.
Well, thank you so much. That was my last question, dear Rector.
Thank you, Dr. Hernandez. The opposition will be continued by Professor Jonkers, Professor of Intestinal Health at Maastricht University.
Thank you very much. Dear candidate, I also would like to congratulate you and your supervisory team with a very interesting thesis. It is a topic which really interests me, and actually both the fibers as well as the polyphenols have my interest, so I learned a lot from them. And also very nice that you combined those complex in vitro studies with the TIM model with very complex in vivo studies. And I have actually a lot of questions I can ask you, but I have to choose. So I will first focus on chapters 3 and 4 on the citrus fruit extract. Where in chapter 3, you first did TIM2 experiments with 2 dosages, 250 gram and 350 gram. And you did then find an increase of, for example, acetate, but a decrease of butyrate as compared to the seam. And then you did go further with chapter 4, and I think it was very nice that you again did a TIM2 experiment with 500 milligrams, and then you suddenly found an increase in butyrate. And then I was a little bit confused, because in chapter 3 I did not find a dose-dependent effect of the 2 dosages, but it both had the same levels of butyrate and both not increased versus seam. And then suddenly in chapter 4 it was. So what are your thoughts about that? How to explain those different findings?
Highly esteemed opponent, thank you for your question. So there is an important factor that I believe will answer your question, is that we use different donors, so it was completely different populations. In chapter 3, we had donors that were metabolically healthy, and in chapter 4, we have the donors for metabolic unhealthy, so people that were in risk to develop metabolic syndrome. So I think that would explain the differences response to polyphenols and the short-chain fatty acid production.
So then it seems to be not so healthy in the healthy volunteers?
I think it's not that healthy, but I believe the good microbiota will not need that push that far, because the amount of polyphenols, if we have like a healthy diet, that was something that we just got from the participants, so they were healthy. So in theory, they were having a healthy diet, so they will have enough fibers and enough polyphenols, so enough fruits, vegetables that will provide enough.
Okay,
food.
but now you trigger me, because I did check the dosages of the butyrate, and then I saw that if you assume that the healthy ones have a higher starting diet with already higher levels of polyphenols, then I would expect that the overall dosage still would be a bit higher then, but the overall dosage is still way lower than compared to the other group. So then I tried to figure out whether that could be an explanation.
That also can be an explanation in the sense that the gut microbiota is already using polyphenols from diet and then was not really using the ones that were available from the supplementation. And I think it would be nice to do maybe a structure in the sense of a structure evaluation in the sense to see how much polyphenol was remaining at the end of the
No, it's for sure interesting to zoom into that. But because of time, I would like to go a bit further on the human intervention study you did there, because I was really very interested in your findings, because I'm very interested in intestinal inflammation and calprotectin. So I thought, okay, this is my chapter, so I can learn from that. So, and then you actually concluded that you found in the abstract, but you also wrote that in your discussion section, that there was a positive effect on intestinal metabolic responses. And actually, my first simple question is, what did you exactly mean by intestinal metabolic responses?
So, in this sense that we just evaluated short-chain fatty acid production and other organic acids and branched-chain fatty acids, that was our metabolic response. So the amount of short-chain fatty acid being produced, but also how much we could inhibit of the branched-chain fatty acid production. And that also was a reflection, I believe, by the modulation of the gut microbiota, because we saw a decreasing of bacteria that are more towards fermenting detrimental.
Okay, so it was more on the metabolite parts. Yeah. And then indeed you came with the remark that the short-chain fatty acids shifted towards more butyrate, which I like because it has anti-inflammatory capacity. But if I then look to page 111, table 4, then I could not find a different effect in butyrate, but I only found a relative effect if you look to the relative amount of butyrate versus the total level of short-chain fatty acids. So I was thinking how to interpret that. I always thought, okay, the absolute level of butyrate is important, but do you then believe that the relative level of butyrate is more important? So how to interpret that finding?
So the technique we use to evaluate short-chain fatty acid production is always, or was always, the cumulative production. So it's always relative. It's not, I don't know how to say that, but it's not like the number per se. So,
but do you agree with me that if the absolute concentration, or in your table there was no significant difference in butyrate, is it then maybe not a bit firm to conclude? How do you feel about that?
Yeah, I think would be maybe I was a bit pushy in that conclusion. But I think I also add to some limitation lines in the conclusion about confirming.
No, but I'm very happy that you see my point. And then I was interested. I mentioned already calprotectin. That would be nice. And also there you maybe can help me how to interpret the findings, because you find a significant difference relatively over time. So I think it's really nice that you did the analysis within a subject, because there can be variation between people. But then I saw that the deltas were in nanograms per ml. And I do a lot of work with IBD patients, and all the cutoff values there are in micrograms per gram features. So if it's below 50 microgram per gram features, then it starts. Then it's normal in IBD patients, so it's in micrograms. and you see differences in nanograms. So I was wondering how to interpret that differences in scale, and do you think it's clinically relevant?
So actually, I believe that was a typo mistake, unfortunately, and we were intending to say micrograms.
Oh, then I'm very happy because that makes a big difference indeed. Then still my question stands, because then the difference is about 10 micrograms per ml, as you indicated here. Do you feel that's clinically relevant, or how should we interpret that?
I think it is. So the population were not technically the most inflamed person, like in IBD. That is really a issue.
Do you know a little bit what the baseline values were about what?
I think for the baseline for the
patients. Yeah, well, how high were their baseline values? Were they considered normal? Were they considered increased?
It was normal, because they were in risk of metabolic syndrome. But I think in this sense, as a protection, it's relevant. Because if they are already in risk and they can respond to increase or decrease the protectin, it's a good thing too.
Yeah, maybe we can zoom into that in a later phase, because I was wondering indeed whether it's kind of in the normal range, yes or no, or whether it really means that there is inflammation. So I was very interested in that. I do not have further time, so that we will do that in another moment. Then I thank you for the answers.
Thank you.
Thank you, Professor Jonkers. The opposition will be continued by Professor Platt, Professor of Physiology of Nutrition at Maastricht University.
Thank you. Yes, dear candidate, I also would like to congratulate you and also your team with this beautiful thesis. It's an important topic. We need a lot of knowledge to think about practical applications, and I think your results have definitely contributed to that. Just like the previous opponent, I also was triggered by the data in chapter 3 and 4 because of you discussed it already a little bit, the contradictory findings. And I was first wondering, why were you so interested in these citrus fruit extracts, so the hesperidin and the naringin? Is there something special with it, or was it?
Thank you for your question, highly esteemed opponent. So there are some studies proving or saying that they have seen in animal, in vitro, and also human studies this improvement in inflammation with people with IBD, but also chronic inflammation as in other populations. So our wish was to use some product that had lots of waste, but also it's kind of common in our diet to provide some benefits.
And of course, you have many different polyphenols. So is this, to your opinion, the most interesting one? So is it because you now present it as a kind of representative for the polyphenols in general, but if you would have the possibility to choose one yourself, is this the most interesting one, or?
I believe this one has a lot of potential based on literature, but I also like that is from a fruit or is from food that we eat in daily basis and we can find everywhere in the world. So not necessarily we would use a supplementation, but we can maybe get a similar results because the amount of the two products that I use as polyphenols, they are based on the natural presence in the fruit. So
and sorry to interrupt, but
yes,
suppose that I have a whole series of different polyphenols, and in chapter 8 you somewhere are quite strong in your conclusions that you think the TIM model is of value in making these screenings. Would you advise me to use the TIM model to choose the most beneficial polyphenol?
yes, I would say so. So TIM2 has its limitations, but I think every single in vitro model will have limitations. So really depends on what you want to answer. But in the case, as I did during my thesis, to use TIM2 as a screening tool to evaluate the response of polyphenols to good microbiota of different populations, or even for one person in the sense of doing a more personalized work, is really helpful, because comparing with other...
Did you do some reproducibility calculations, for example?
No, I didn't. So we believe that because TIM2 is computerized, it keeps the reproducibility quite well. But as I mentioned before, doing triplicates would enhance our certainty about the results. But so then you have the limitation of a budget when you run. So you have to weigh
you would advise
what you...
at least to do the duplicates. And I was triggered a little bit by the question by the previous opponent, because there you answered that the TIM is always a cumulative effect. And then transferring the data to humans, there you have a one time point. But that's also the concentration within the lumen, of course. So would it make a difference, suppose it would be possible to sample maybe in the portal vein or somewhere more in circulation? Would that make a difference? Does it accumulate somewhere?
So you say if we take samples from TIM2 in another way?
No, in the human situation.
In humans, okay. I believe would make a difference indeed, because we have the epithelial cells absorbing those metabolites, or what is remaining from the polyphenol. Polyphenols that reach the column, so I believe it would be interesting to compare and see where the polyphenols are going in our body and what exactly is being affected in this sense.
Okay, and then looking at all the chapters together, so to say, so you tested all these ingredients, let's say one by one, in controlled interventions. What about a combination of all the different compounds? What do you expect? Will they have kind of additive effects, or maybe synergistic effects, or maybe even that they contradict each other, because one compound might influence the bioavailability of another, or what's your expectation?
So it's something that I'm thinking about of doing, because I believe the matrix of the food will influence the availability of those components. So that's why I also highlight quite often during the thesis that evaluating the structure of the food would be interesting. But I wish I can in the future do media that mimics a whole plant-based diet, and then we can see and compare with the SIM that mimics the westernized diet to see what is the response, because so far it's going to be complicated to analyze and know what is affecting what, but what we eat at the end is a complex range of foods.
It's very complex, yes. So what would be your, let's call it your Maurer Sost diet? What would the diet look like? Is there sweeteners in it? Is there polyphenols, fibers, which maybe even things you did not test yet? So what would your ideal...
So I believe eating lots of vegetables, and when I'm a dietitian and nutritionist, so I have been working with people that want to lose weight and people with obesity quite long, and because of the culture of losing weight, people are afraid of even eating a salad. So when we, as health providers, we indicate that person, okay, maybe we can change your diet. I believe the first question we should ask is, what do you like to eat? And goes from there. And then within that wishes and likes and dislikes, we reach the point where we can introduce new things, so more fruits, more vegetables to that person, so to that person diet, so we can reach this amount of dietary fibers that we in line with the recommendations, but also providing this energy for the good microbiota so we can have these results that we see in the individual basis.
Okay, so we need some research for that. I think I agree, and I'm very happy to hear that you think we can do it with normal foods and we don't need all kind of supplements and those kind of things. So thank you for the answers, and I give the word back to the prorector.
Thank you, Professor Platt. The opposition will be continued by Dr. Zoetendaal from the Laboratory of Microbiology at Wageningen University and Research. Thank you for coming to Maastricht.
Thank you very much, Prorector. Dear candidate, I also would like to congratulate you and your supervision team about this beautiful thesis. Nice work, a lot of chapters, something you can be really proud of. And I read it with pleasure, and I would like to ask some questions about it. First, the cover has already been briefly explained, but there is in the cover also the word functionality, and that can be broadly explained. So can you give your explanation of functionality?
Thank you for your question, esteemed opponent. So the functionality during my thesis was about the way how and how much the bacteria could produce short-chain fatty acids and other organic acids, even the detrimental ones.
So it's mostly about the fermentation?
Yeah.
Okay. Can I ask one of your partners to read Proposition 7?
Which one? 7.
Detailed characterization of both microbial and metabolic phenotypes, including their interaction, is necessary to improve understanding of the variability in individual responses to dietary interventions.
Yeah, thank you very much. Yeah, interesting word there is individuality. And when I look at most of your chapters, except chapter 7, you look at pooled samples. So why did you choose for pooled samples and not individual samples?
So I think the personalized concept is pretty new. And what we saw over the years was that maybe the fecal sample that was pooled was not answering the questions we were asking. So what we see with the fecal sample that's pooled, we can represent in some way a population. But what we have seen in literature nowadays is that we have people that respond to treatment and people that doesn't. So we thought that would be interesting to see if with Team 2 we can trace this and may use Team 2 as a screening tool for that. So although most part of my thesis is with fecal samples, we thought that would be interesting to see if we also can get this prediction or these differences in response depending on the gut microbiota of a unique person. Okay, yeah, that triggers me a bit because you concluded based on the chapter 2 that the asparagus substrate preserved or improved richness and community diversity in the overweight and type 2 diabetes risk group. But what if I give you this conclusion that I made myself on this chapter? I looked at the inocula. You have a pooled one and you have a healthy one. One of each. One contains 7 fecal microbiomes, other contains 8. The healthy contains 8, and I concluded, like, well, you start with much more fecal sample, much more bacteria, so you have a higher diversity, and this can, just the fact that you already have a starting different diversity, explain the differences, and not the fact that they are obese or healthy. Can you argue about it? Yeah, that's a good point. So that might have a difference indeed because we have one more microbiota population there that might not match with the other donors. But I also believe that there is some overlapping functionality in the sense. So although maybe the initial microbiota is important, we also to translate the good microbiota importance is to see what comes out of it. So what's the fermentation pattern, are the outcomes from that fermentation. So the function of the bacteria, they overlap. So maybe I don't have the same bacteria that you have, but we can reach the same outcomes.
Yeah, that could be, but I was also wondering because if you look at the different chapters, you use different pools of, for example, the healthy ones. And actually, one of the questions I have is that why you choose at one time 7 or 8 or 11 subjects? Is there not a standard? But have you also compared the controls with each other? We have only seen, and do the pools represent something very similar, or is it like the individuals that it goes in all directions?
Sorry, could you repeat?
Sorry. If you have the control samples, the control pooled samples of each chapter with theme, have you compared those to each other to see how similar they are?
I believe due to time frame of the studies, they might resemble each other, but they are not going to cluster together if we print it as a PCA graph, because they are from different donors.
Yeah.
And the time that we did some experiment with the time that we did further in the other chapters was quite apart, maybe even some 2 years. So I believe we're gonna see differences in that sense.
Yeah. And why do you choose in one study 11 subjects and in another study 7? Is that because one person produces more in the toilet than the other, or what's the reason?
I have to say that, yes, might be a reason. But I have to say, and you probably know because I also work in the area, that's quite difficult, and maybe asking for fecal samples to people, it's quite a taboo. People get like shy, or I don't want to do it, and yeah. So I think the study that we did during with the 7 donors was during COVID time. So approaching people to donate fecal samples was much more difficult than later times. So that's, I think, is the biggest why we had the differences.
Dear Prorector, do I have still time for some questions? Yeah, then...
You have another question? There's time.
Okay. Then I would like to ask some questions about Chapter 6. First question is, did you submit it to Nature?
Yes.
Yes?
Yes, was accepted, but we are not published yet.
Not published yet.
Yeah.
And was it because, congratulations if it's going to be accepted, but was it because of the methane-producing bacteria that you found?
If it was accepted.
Was that the most important reason why it is submitted there?
Yeah, so actually it's the first long-term clinical trial that use that high number of participants to show the effects of having the intake of sweeteners or not.
Yeah.
So I think the magnitude of the study was the reason why.
No, I'm asking that because I was really intrigued by the fact that you described you found a methane-producing bacterium. Is that correct?
Yes.
Was it a bacterium?
Yes, so we also, for the Nature, we went further in the results about the gut microbiota to discriminate responders and non-responders. So we saw a bit more specific changes in gut microbiota in this sense, but I didn't include these results in my thesis back that time. Okay,
because I think it was Methanolobus you found. Is it a bacterium or is it an archaeon?
I believe it's a bacterium.
Bacterium, okay. I thought it was an archaeon that you found, but if it's bacterium, it's very interesting, and then it would be great to isolate it. But was a bit intrigued because I've never or hardly heard about methane-producing bacteria, only from the archaea. So that's why I was wondering if this was really a bacterium or not. But maybe check later that part. And I think I'll leave it here. And yeah, thank you again and give it back.
Mônica Maurer Sost, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
All right, please.
Mônica Maurer Sost, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Blaak is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
All right. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Mônica Maurer Sost, the degree of doctor and grant you all the rights attested by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university. So, dear Mônica, I'm the first to congratulate you on behalf of Team Mônica. I joined your journey during the last years, and I think you made great progress and you did a great job today. So be proud of it.
Thank you very much.
I'll pass the word now to Kalili, who will do the laudatio.
Thank you, Professor Ellen. There you go. Ladies and gentlemen here present, for everyone who also joined online, and especially her sister, her newborn niece, and her parents probably following from our home country, Brazil. I will have some words in Portuguese because, yeah, we are Brazilian, so baguria. You came from so far away, carrying with you your culture, your warmth, and your kind heart. You built a life here and with your husband, one step at a time. You created friendships, contributed to science, and inspired me with your empathy, happiness, and strength. Mônica and I have some things in common, as I said. We are both from the south of Brazil, and we share the same bachelor degree, right? And we had this dream of working abroad and do nice research in the gut microbiota field. I believe that's how destiny, or maybe God, brought us together. For these reasons, and many more, I'm truly happy for you and for your achievements, of course. And I'm also here to say how glad I am that you were here. Mônica helped me a lot when I was moving to the Netherlands, and she kept helping me along the way as a work colleague, but also as a friend. Knowing your journey to this point, it would be impossible not to be happy for you. Today I want you to know you made it with all the ups and downs. You overcame all the obstacles and you made it. Mônica has many wonderful qualities, and one of them is empathy. She has an incredible ability to see and care for others. And alongside her empathy, she's very, very intelligent. And there's something even more special about her, that is her courage. To everyone here, do not underestimate the strength and power of this little lady. She might be small, but her bravery and courage are much bigger than she is. In our group, she was always ready to speak up when something felt unfair or when she saw things that were not right. She was never afraid to stand for what she believed in, not out of confrontation, but out of deep sense of integrity and justice. Her strength inspired me. not to be afraid to speak up when things were not right. At work or outside, we had countless moments of fun together, at conferences, dinners, cycling to work or playing video games, trying to beat levels in Super Mario. Do you remember that? We also had many nice times with Judy and Nohaila in the lab in Maastricht with Yuri as well. We all know that a PhD is not only about science, it's also about resilience, hard working, and those moments when you wonder if it will ever reach an end. For example, when performing full experiments in Team 2, like full day experiments, and when the incredible machine was not so incredible and had some issues. Or struggling with so many results, statistics and R, for example. And to add here, why did we choose to add, as Alex used to say, why didn't we choose to work with flowers, right? They smell much better. She learned everything and handled them mostly by herself. Things got even harder with the difficulties and uncertainty times we faced in our group. It caused us a lot of stress and many, many hours overthinking and over-talking. Still, gladly you continue, and believe me, those difficult moments were easier to bear because of you. But things became much better when Professor Ellen and Emmanuel began to supervise her, and I have to thank them, you, Ellen, for your help and your support. You put a lot of effort in Monica's journey. Thank to you for all of your help. I say this also on behalf of Monica. I believe she's really helped that you joined the group at some point. And of course, I have to say there's something else about Monica that most of the people don't know, and I cannot forget. I cannot forget one of Monica's fun and charming qualities. She is incredibly photogenic, looking joyful and full of life in every picture. So today, please shine to your pictures to make this remarkable day even more special. Monica, congratulations. You really deserve this. You made everything you could to have this day. With all the ups and downs, with all the problems, you made it. So, parabéns. Parabéns, doutora. You truly made it. Congratulations.
Thank you.
Yeah, dear Dr. Maurer Sost, also on behalf of Maastricht University, I would like to congratulate you with the degree you have acquired. It's an important day. And I hope you really enjoyed the defense. Yeah?
Yeah,
You did?
I did. I did.
That's a good
More
sign.
than I thought I would be.
Of course, I also congratulate your family, friends, and colleagues present here or online that have watched this ceremony. I'd like to congratulate your supervisors with this PhD defense and thesis. It has been hard work for you as well, I understand, but it has come to a really good end. Thank you for that. I also like to thank the members of the degree committee, and especially the members from outside Maastricht, Dr. Hernandez and Dr. Soetendaal. Thank you for being here, and thank you all for the time you have invested in preparing for this meeting. Maastricht University really appreciates that. Yeah, I think we've now come to the end of this ceremony, so I close this academic session.
Thank you.